the current situation in the United States, but also globally. And then last but not least, I'm going to take a look at the serological aspects of measles and mumps as far as the diagnosis is concerned. So I would like to start with the measles virus. So the measles virus as such belongs to the family of the Paramyxoviridae and belongs to the genus of the morbillivirus. And so far, 24 different genotypes have been described. The virus has an envelope and a single-strand negative sense RNA genome. And as a matter of fact, the measles virus is one of the most contagious human diseases there are, and they are usually spread through respiratory droplets. Infections usually generate this very characteristic skin rash that many people have already seen, and there is no treatment available specifically targeting the virus, so every treatment is tailored towards a systematic treatment. Taking a look at the numbers on an annual basis in the United States as far as infections or reported infections in the United States. Back in the 60s, we were dealing with around about 500,000 cases on an annual basis. And ever since the vaccine was licensed and introduced in the 60s, there was a dramatic drop, a dramatic decrease in numbers of infections, with sometimes peaks, for instance, in the late 80s, early 90s, also leading to a recommendation for a second shot of the vaccine, which certainly contributed to the fact that in 2000, the disease was declared eliminated. However, there are still some localized outbreaks throughout the United States, ranging, for instance, from 37 cases in 2004 to as high as 667 cases in 2014. But it's not only a US-specific challenge. As a matter of fact, measles outbreaks still occur on an annual basis globally. And this is what we're looking at right now, basically a heat map of 2017 and the outbreaks that were reported on a global scale. And it still is true that most of the cases or reported cases of infections were identified in uninfected people. And oftentimes it's travelers who bring the disease to the United States or Europe. And certainly in days with a refugee and immigration challenge, this also increased the problem on a global scale. Next, I want to talk about measles serology as far as the diagnostic component is concerned. So molecular testing is a major cornerstone of especially the acute diagnosis of a measles infection. However, serology can play a very strong supporting role in discriminating between acute and past infections, but can also help identify neurological manifestations that I'm going to talk about at the end of this part. So let's talk about serology and its use in the diagnosis of acute infections. So obviously, the clinical symptoms have to be in place, molecular testing hopefully as well, and combined with the serological results, a diagnosis for an acute infection can be made. What do we usually observe in patients suffering from an acute infection? So the IgM antibodies are normally detectable within the first days after the appearance of the skin rash. But even in IgM negative patients, you cannot rule out an acute infection for a number of reasons. IgG can also be a very powerful tool to diagnose or support the diagnosis of acute infections. In paired serum samples taken in an interval of several weeks, acute patients usually show an approximately 4-fold titer increase for IgG, indicative of an acute infection. And last but not least, testing for avidity for IgG can also help differentiate between a primary acute infection and the previous or past infection. And the avidity testing is something I would like to talk about very briefly on the next slide. So the basic concept is that in an acute primary infections, in the early stages, patients will have low avidity antibodies that are somewhat unmature. And if they mature throughout over time, they will become more and more high avidity antibodies, which can be challenged or can be detected with specific immunoassay setups. So in an immunoassay setup, we usually use urea because low avidity antibodies when challenged by specific UR concentrations, will lose their bond with the antigen presented, for instance, in an ELISA format. However, high-avidity antibodies will withstand that stress put upon them through the urea incubation and will maintain the bond with the antigen. Thus, this can differentiate between high-avidity and low-avidity antibodies and can help differentiate between acute primary infections and past infections or reinfections, as well as reactivation of the virus. Another aspect that I think is worthwhile talking about is a potential link between past measles infections and the neurological manifestation that is referred to as subacute sclerosing panencephalitis, which is a generalized inflammation of the brain with severe nerve demyelization. And it's hypothesized that this might be caused by persistent measles virus infections in the central nervous system, a kind of slow virus disease, potentially caused by mutated viruses. In patients, you usually observe extremely high titers in the CSF, the cerebrospinal fluid, for IgG antibodies against measles. And oftentimes, those manifestations occur about 7 years after the initial infection. And the group that seems to be of highest risk to develop this neurological manifestation is kids that had their measles infections within their first year of life. In Germany, between 2007 and 2015, 280 cases were reported for SSPE, which accounts for about 2% of all cases with an acute infection. Unfortunately, there is no cure available for SSPE, so that means prevention in terms of vaccination still remains the one tool that can prevent that disease. From a commercial perspective, I would like to introduce a couple of ELISA concepts, knowing that for IgM and IgG testing, there is also IFA solutions available from Euroimmun. Starting with the IgM ELISA, most prominent product, anti-measles NP ELISA based on the recombinantly expressed measles nucleoprotein, the major target of early immune response, hence paired with the IgM. The IgG ELISA, since there is international standards available, is calibrated as per the international unit and comes with a 4-point calibration curve that has multiple utilities. The avidity testing is available through Euroimmun as well. It's pointed out to discriminate between high and low avidity antibodies. And last but not least, it requires special tools, special assays to detect the very high concentrations of IgG antibodies against measles in CSF. And also for that application, we have an assay that is commercially available. Moving on to mumps. There's quite a bit of overlap between measles and mumps. First and foremost, they belong to the very same family, which is again the Paramyxoviridae. In contrast to the 24 genotypes that have been described for measles, there's only 12 different genotypes described for mumps. Again, an enveloped virus with a single-strand negative RNA genome. Also spreads through respiratory droplets. And infections usually cause this very characteristic swelling of the salivary glands, and in male patients, sometimes testicular swelling can be observed, which fortunately only rarely results in infertility in teenage males and men. It's accompanied by fever, headache, and rash. And also worthwhile mentioning is that about 15% of infected patients develop a form of meningitis. Similar to what we saw for the measles, there is a very fortunate trend that we see in the United States also for mumps. In the 60s, we were dealing with around about 150,000 cases per year, which then dramatically dropped after the vaccine was introduced to the market, and in 1977 an MMR combination vaccine was released, which improved things even more. There was in 1993, 1994, just for measles, the Vaccine for Children program, which allowed children or school kids to have access to the vaccine even without health insurance, which improved things dramatically. But there was one major outbreak in 2006, mainly localized at universities, not very spread out far, but mainly affecting young adults. Looking at the most recent data put out by CDC, we can see something that might be a little bit disturbing in a way, because 2016, 2017, we saw again a dramatic increase in numbers, close to the 6,000 reported cases on an annual basis, which pretty much is what we saw in 2006. So currently there seems to be an increase again in cases of mumps for the United States on an annual basis. Something I would like to introduce at this point, which is not only of importance for the serology, but also something Rolando is going to talk about later, is the phylogenetic tree of the mumps virus. Because there has been a shift in what we observe as a wild type virus causing the disease. So back in the day, the genotype A was the predominant wild type, with the Andes being the specific strain causing most of the infections. However, in the recent days, we saw a shift to more of the G genotypes causing the vast majority of infections, especially in Europe and the United States. This is backed up by data put out by the WHO that specifically show that the G genotype, and specifically the G5 strain, are the cause for the vast majority of cases in A, Europe, but B, also the United States. And this led Euroimmun to develop a new test system that focuses on the G5 as an antigen for their IgM Eliza solution. And again, just like for measles, the molecular testing plays a major role in the acute diagnosis of infections, but just like for measles, in the mumps case, ELISA solutions and IFA solutions in terms of serology play a very strong supporting role when it comes to the diagnosis of the disease. So what was the advancement switching from Andes to G5 as an antigen for our ELISA solutions? So what you can see here right in front of you is that the switch from the Andes strain to the G5 strain significantly improved the signal-to-noise ratio compared to the old standard, which led to a clearer discrimination between positive and negative sera. This means an adaptation to what we see out there and use that as an antigen significantly improve the performance of the ELISA that we promote. Another aspect that I want to talk about, which is also related to the different genotypes, is a development that is touching an IgG ELISA that we developed. So IgG, just as we saw it in the past for measles, can be used to support the notion of an acute infection when there is a fourfold titer increase over a couple of weeks detected with the paired samples. But on top of that, combining the wild type strain, the Andes, with a vaccination strain, the idea was that potentially we can also improve that assay as far as the detection of immune response in people, or especially children, right after vaccination was concerned. And to test that notion, we pretty much compared the wild type strain ELISA only with the Andes antigen to the combination of the wild type strain, and added the vaccination strain, which is the Gerald-Lin. And those two tests were compared in different cohorts, including children after Mmrv vaccination, adults after vaccination, and adults with former infections with a wild type virus, as well as negative controls. And in this internal study, you can see that those two assays performed similarly well in adults after vaccination or after infection, past 100 days, at 120 days. But what we can see is that by adding the Gerald-Lin strain to that ELISA, we saw a dramatic increase in the sensitivity as far as the detection of an immune response is concerned in children less than 60 days, 60 to 120 days, or 120 days after the infection. It is important to note that at this point, those antibodies that are detected by the ELISA basically represent an immune response, but do not necessarily have to correlate one-to-one with immunity or neutralizing antibodies. Last but not least, the small cohort with the controls did not pick up any additional signals or still maintained negative with the new ELISA. So in short, this ELISA can be used to detect or support the notion of acute infections by means of the titer increase, and it has an increased sensitivity in early phases after vaccination of children. So with that, let me sum up my part. I talked about diagnostic tools for acute diagnosis, but also neurological manifestations. For measles and mumps, there is IgM and IgG tools available, both ELISA format and IFA format, indirect immunofluorescence format. For measles, there's also an IgG avidity assay available that it talks about, and a specific test kit that can detect intrathecally produced IgG antibodies with a specific test kit. There's also an assay available that works with mumps viruses and IgG CSF, however, I've not talked about that. With that, I conclude my part and basically ask the question, how can we use some of that knowledge and additional knowledge and ask the question, okay, what's actually going on in terms of a vaccine perspective? And with that, I would like to hand it over to Rolando from Covance to talk about more about these aspects.
Thank you so much, Oliver, and thank you all the audience for attending to our webinar today. As it was introduced by my colleague from Euroimmun, the vaccines for the prevention of measles and mumps had a dramatic effect of reducing the incidence of these viral infections. The current vaccines are live attenuated. That is, they contain a live weakened virus and are also combined either rubella in the Mmrv vaccine or rubella and varicella in the Mmrv vaccine. Different formulations of these vaccines were investigated during the development process to obtain acceptable immunogenicity and safety profiles. As a result, these vaccines produce a subclinical or mild non-communicable infection, inducing both humoral and cellular immunity. They have been widely used in the US, Australia, Canada, in many European countries. They are manufactured by different companies, mainly by two, GSK and Merck, and they have a slightly different composition. Licensed Pro-Quad and Priorix Tetra vaccines have different measles strains, Edmundston and Schwartz strains respectively. They do have, however, the same titer, expressed as tissue culture infectious dose, TCID50. The mumps virus in strain in Priorix Tetra, RIT4385, is derived from what it is used in the Pro-Quad vaccine, which is a Jerry Ling strain. Both vaccines have the same rubella virus strain, the Wistar RA273, and titer. And they also have the same varicella strain, the OCA strain, but with different titers. Both vaccines have performed similarly in clinical evaluation and achieve equivalent efficacy levels. Thus, it is not a surprise that the CDC position regarding the use of these vaccines and their effectiveness is the following. A single dose of the Mmrv vaccine will confer 93% of protection against measles and a 78% protection against mumps. A two-dose of the vaccine will be 97% effective for measles and 88% effective for mumps. About 3 out of 100 people who get 2 doses of the Mmrv vaccine will get measles if exposed to the virus. However, they are more likely to have a milder illness and are also less likely to spread the disease to other people. While 2 doses of the Mmrv vaccine are 88% effective at preventing mumps, mumps outbreaks can still occur in highly vaccinated US communities, particularly in close contact settings such as schools, colleges, and camps. High vaccination coverage helps limit the size, the duration, and the spread during these outbreaks. However, if we say these vaccines are effective, how come we still get localized outbreaks? To understand that, let's dive into how these vaccines work, mechanistically speaking. Make no mistake about it, antibodies are very important in mediating protection against measles. For example, we know that either a gamma globulin treatment or maternal antibodies protect against the disease. Antibody-based functional in vitro assays also correlate with vaccine protection. The best example is perhaps that microneutralization values of 125 milli-international units per ml give protection against disease, while a higher value of 1,000 milli-international units per ml protect not only against disease, but also infection. There is also evidence of the role that cellular immunity may play in the protection. An early example was captured by Badara-Sand and colleagues in a study published in 1995. They looked at the antibodies among vaccinated and unvaccinated 12-month-old children. They found that both unvaccinated and vaccinated children with plague neutralization titers of 40 milli-international units per ml or less were highly susceptible to clinical disease. They also found that immunized and unimmunized children with titer of 125 milli-international units per ml showed a high degree of protection. Unvaccinated children, and this is where it gets very interesting, with antibody levels of 40 to 125 milli-international units per ml, still had a high risk for measles. But vaccinated children with antibody levels of 40 to 125 milli-international units per ml were protected. One interpretation of these results is that only immunized children were not protected by the maternal antibodies, whereas immunized children had cellular immunity in addition to the antibodies. This is an indication of the role of a cellular response in protection against measles. If infection does take place, T cells of both arms must be functional to close off replication, which is why T cell deficient children may suffer from complications following vaccination. In animal models, whereas CD8 positive T cells are mainly responsible for viral clearance in the lungs, only CD4 positive T cells seem to be necessary for control of viral replication in the CNC. In summary, by using solely the antibody levels as a correlate of protection, we could be underestimating the protection level elicited by the measles vaccine in certain individuals. This becomes more of an opportunity when we think about personalized medicine. Developing novel correlative protections for measles could lead to a new option in the rapidly expanding menu of personalized immunity. The reasons why we still get measles cases come down to importation of disease from areas where the vaccine is still not widely used, and low vaccine coverage among certain population groups. It could also be due to waning immunity or discrepancies to circulating strains. Having said that, the measles vaccine is effective, and it seems to be as good as it gets. Mumps, on the other hand, is a very different story. There is no clear correlation of protection against mumps. We know that antibody transfer does not protect. We also need to keep in mind that different mump vaccines may contain different strains. They elicit different Eliza titers, and while they elicit similarly neutralizing antibodies, the efficacy levels may be slightly different. Furthermore, we know that passive immunization, and that is a transfer of antibodies, do not confer protection against mumps. It has been suggested that titers of 1 to 2 by neutralization or 1 to 8 by heme agglutination inhibition could be a correlate of protection. But we also know that while T-cell responses do happen following vaccination, their protective role currently is unknown. The bottom line is that mumps outbreaks in previously vaccinated young adults who apparently lost prior immunity still happen and highlight the need for a defined correlate of immunity to mumps. The mismatch between the vaccine strain and the currently circulating strain has grown bigger, as Oliver explained during his presentation. And it is shown very clearly in this unrooted tree. The vaccine strain Gerylin on the right belongs to a rather different genotype from the most common isolates in the Western Hemisphere, which are G genotypes. And to further highlight the impact of strain diversity in the potential efficacy of the vaccine, I would like to discuss a publication from 2015 in Pediatric Research. In this publication, Sakara and colleagues studied 281 children with what they define as mumps reinfection. Of them, 240 had a natural infection, a primary mumps infection as the author defined. 9 had a reinfection following the natural infection. 26 had mumps despite being vaccinated at some point in their life. And there were 6 cases of vaccine-associated mumps. When looking at children with primary mumps and mumps reinfection, things got very interesting. Among those children infected following a natural infection, 7 were IgM positive and 2 were negative. And that's a clinical serological diagnosis. However, among those that acquire mumps despite being vaccinated, 50% were IgM positive and 50% were negative. This is a clear indication that serological diagnosis is affected by the immunization status conferred by the current vaccines. It supports the efforts of Euroimmun in adding a new strain to their mumps IgM kit. And it also points towards the need to use quantitative IgG levels as potential biomarkers of infection and immunity among vaccinees. It is also worthy to note that all but 6 isolated mumps virus from 41 children were of the more common, not vaccine-related genotype G. Again, highlighting the effect of the diversity among the circulating strains. This mismatch as a factor in mumps infection is also apparent from the data on the neutralization levels of serum from 96 different individuals against 7 different mumps strains. As we can see, the highest neutralization titer was achieved against the Measles vaccine strain, and the titers decreased from there as more divergent strains were tested. Conceivably, we should also expect that this neutralization activity may decline with time following vaccination. Thus, it is not a surprise that several authors have studied the decline of anti-mumps antibodies and anti-measles and anti-rubella antibodies following MMR or Mmrv vaccination. To that regard, Siegel and colleagues published their findings upon a 12-year follow-up to the MMR vaccine among vaccinees. They found that most of 302 participants were seropositive throughout the follow-up time. 1 month post-vaccination with the second dose, however, the geometric mean titer to measles was high, but declined on an average of 9.7% per year among those with the same baseline titer. Geometric mean titer to rubella declined between 2.6 and 5.9% per year among those who received the vaccine at different time points. The geometric mean titers to mumps declined at about 9% per year, regardless of their demographic or age. In another study, this time among vaccinated students in the University of Iowa, Cardamil and colleagues found that the mumps attack rate was 6.7 per 1,000 among students who received a third dose of the MMR vaccine, versus 14.5 cases per 1,000 in those who didn't. At 20 days after vaccination, receipt of a third dose of the vaccine was associated with a 78% lower risk of mumps than those that received only a second dose. As compared with students who had received the second dose of the MMR vaccine within the previous 2 years, those who had received it 13 to 15 years earlier had 7 times the risk of infection, and those who had received it 16 to 23 years earlier had 11 times the risk. These findings suggest that the campaign to administer a third dose of MMR vaccine improved mumps outbreak control, and that, more importantly, waning immunity is contributing to the propagation of the outbreak. In a different study from the end of July through September 2017, Gent and colleagues found a total of 26 cases of mumps that occurred in a company or 140 military recruits. Among all the soldiers, 91% had received 2 doses or more doses of the MMR vaccine before the outbreak at some point in their lives. And the overall attack rate for the entire group was 18%. As a follow-up to the outbreak, they investigated whether the administration of a third dose of the MMR vaccine had been protective by stratifying the data according to the timing of vaccination. In line with the report by Cardamil and colleagues, the attack rate was 86% among soldiers who received no vaccination. It was 15% among those who had received 2 vaccine doses, and 8.3% among those who had received 3 vaccine doses. More importantly, no mumps cases were reported among the soldiers who had received the third MMR dose in the year previous to the outbreak. But the attack rate was 14.3% among those who had received the third dose 2 years before. Although the difference in the attack rate was not statistically significant, and mostly due to the sample size in this study, these findings suggest that the timing of the administration of a third MMR dose may be important. Data from a third study by Weber and colleagues also appears to agree with Cardamil findings, in the 13 years after MMR vaccination, there is a sharp decline in mumps seropositivity. All these findings have led to a change in the ACIP recommendation that was published in January this year. The current routine recommendation for 2 doses of MMR vaccine appears to be sufficient for mumps control in the general population, but insufficient for preventing mumps outbreaks in prolonged close contact settings, even where coverage with 2 doses of MMRV vaccine is high. Waning of vaccine-induced immunity with time after that second dose in high-intensity exposure settings contributes to these higher risks for mumps disease. Protection against severe disease, however, is maintained, but considering the evidence regarding the public health burden of disease and the known risk factors, persons who are at increased risk for acquiring mumps because of an outbreak are identified as a public health priority for receiving a 3rd dose of a mumps-containing vaccine, either MMR or MMRV, bringing the vaccination with the MMR and MMRV vaccines back to the spotlight. In summary, measles and mumps remain extremely important health challenges, especially when vaccine coverage comes down and immunity wanes with time. The new circulating strains, the variability, and the new insights that we are finding trigger innovation of commercially available serology diagnostic assays. The development of new vaccines against these viruses should be matched by a better understanding of the mechanistic elements of our immune response and what is protection and what confers protection against infection and against disease. And that's why it's so important that companies like Euroimmun and Covance are stepping up their joint efforts and provide support to ongoing development to new novel vaccines and diagnostic tools. And with that, I want to thank you so much for attending to our webinar, and we are open to taking questions from the audience.
That's right. Well, thank you both very much for your insightful presentations. As you said, our audience members are welcome to continue sending in their questions or comments using that questions window for the Q&A portion of this webinar. And I've already received a few questions, so let's go ahead and get started with those. Our first question, can the immune responses to measles, mumps, and/or other MMRV components be used as biomarkers of overall immune health?
Yep, and this is Rolando here, and I'm gonna take that question head on. It is a very interesting point. Certainly, the immune response to these viruses can be used as a biomarker to assess the overall immunity status of a person. And it has been in a couple of papers that I can remember. For example, in Hawker, in a very recent paper in Pediatric Nephrology, MMRV immunity was used, was monitored during rituximab treatment in renal transplant patients. And it was used precisely to try to understand how the treatment was affecting their ability to mount a protective immune response against very common circulating viruses. And another application of MMRV testing that where antibodies to MMRV are used as a biomarker comes from the intrathecal humoral response to measles, rubella, and varicella, which is called the Mmrv reaction, which right now is actually the most specific laboratory marker for multiple sclerosis. And I can suggest the audience to look at a paper by Jarius et al. In Journal of Neurology, published in 2017.
Great, thanks very much for that answer. Our next question, how does the nucleoprotein compare to cell lysates as antigen sources to detect IgM antibodies against measles virus using Eliza?
Hey, I think this is Oliver. I think this is a question I should take. The nucleoprotein, and this pretty much reflects our idea for the IgM test for the measles, in which we use recombinantly expressed nucleoprotein compared to lysate-based native antigens. The idea behind that is to have a known target that is specifically the target for the early immune response, namely the IgM response. And by eliminating factors that are not the primary target of IgM response, and by only having the nucleoprotein in a high concentration as the single antigen in the ELISA, we basically achieve two things at the same time. The first thing is we increase the sensitivity and we even increase specificity. I mean, that's something from an immunoassay perspective that you can only dream of. And this is exactly the scenario: higher sensitivity, higher specificity by choosing the primary and first IgM response target. Yeah.
Great. Another question here. What has to be considered for CSF diagnostics of measles?
I think, Rolando, I'm going to take this one as well, because it's basically directly talking about one of our assays, SSPE, as a manifestation or potential manifestation of measles infections. CSF diagnostics is something that should be done in a specific way. You always need to have paired samples of serum and CSF. And preferably, or as a matter of fact, as a prerequisite, those have to be sampled within a very short period of time. So oftentimes you recommend that it should be done within 1 hour that you draw CSF and blood, because there's a calculation involved that ultimately will confirm or will not confirm that the patient intrathecally produces IgG antibodies against measles. So that's the first thing. The second aspect is that you need to use specific assays for this application. Because the sample is different. We use CSF as compared to blood, means the matrix is different. The assay has to be optimized for that. And secondly, very important, especially in the case of measles CSF diagnostics, that the assay has to have a very broad range of detection, because the titers of the IgG antibodies in CSF are extremely high as compared to the titers we see in serum. So it is A, the sampling, and B, using specific kits for this application, in short. Thank you.
Great, thanks for that response, Oliver. I think another one here for you. What are the specific antigenic targets for the anti-measles IgG that are utilized in serology testing?
All right, the IgG test is based on native antigens that are derived from lysates from infected cells. So native antigens, whole lysate from infected cells.
Perfect. Another one, does the lack of cross-reactivity between the Enders and Gerald-Lynn strain translate to neutralization assay?
Maybe I can start this off and ask Rolando to add more to that. I think that's a pretty complex question, and I would like to break it down a little bit more, because generally speaking, cross-reactivity in an immunoassay does not mean or necessarily mean cross-protection. I think that's one important point, and that antibodies, especially in the mumps scenario, that can be detected with an immunoassay, do not necessarily have to be neutralizing characteristics. That's why at the end of the day, in order to determine whether those antibodies are neutralizing, the neutralizing assay is still super important. Also for mumps, the challenge is what Rolando also talked about in detail, is the switch from a PAS predominant wild type to the G, or specifically the G5s, causing the infections, which makes it very hard to come up with an international standard, which we, for instance, see with the measles, where we have an assay that is aligned and calibrated as per the international units and can, as a matter of fact, potentially be used to determine immunity. That is not the case for mumps because of the diversity of the strains causing the infection. And last but not least, I think some aspect that we have not really, I have not talked about, but Rolando touched upon, is the fact of cellular immunity, cellular response, which we do not address at all in an immunoassay of that design. So it is very hard to answer that question because of all those different aspects that play into that. Rolando, is there anything you would like to add to that question?
No, I think you have covered the main points. The heart of it is that any cross-reactivity that you see in an ELISA format, as you mentioned, may not necessarily correlate with a neutralization assay, which is what actually correlates with protection, with efficacy shown by the measles vaccine. It's not necessarily ELISA, it's a neutralization titer of 125, what is considered an international cutoff value for defining immunity. And it comes down to, in an ELISA format, you're detecting you're taking global view of all the antibody response to the virus, while only a subset of those antibodies will target specific neutralizing determinants on the surface of the virus. So that leads to a lack of correspondence or correlation at time between what we see in a traditional ELISA versus the more complex neutralization assay. So, and everything else you add, Oliver, is a fantastic way to address this question. Thank you.
Thank you.
Thank you both for weighing in on that one. Another question for you, Rolando. What are the antigenic targets of the protective or neutralizing anti-measles IgG?
Well, measles is an enveloped virus. So by definition, antibodies that will target envelope and the different proteins that are in this envelope are potential target for neutralization. It is, I would say that the most typical and known target is a hemagglutinin. It's similar to what we hear in the flu side, in the flu virus. There is a test that we can run that is called hemagglutination inhibition. So measles, it's also a virus that can be tested that way, because hemagglutinin is definitely a target. Specifically, the C terminal portion can be a target of neutralizing antibodies. But there are other targets, and there are different groups that have gone through very interesting approaches for mapping all the neutralizing epitopes that can be in the measles virus. And I will happy to follow up on some of the papers that where those epitopes and approaches are described using NGS, using novel immunoprofiling approaches. So I would encourage the member of the audience to reach out via my email for more specific answer on that.
All right, our next question here. Can you confirm the rationale for calling for a new measles vaccine, considering outbreaks have only been associated with poor vaccination coverage?
Right, so definitely, and that's a question for Rolando, me. I'll take it, only review mine. It is true. It is true that the current measles vaccine works. But as you can see for the data, it seems to work for only a fixed period of time. And that raises then the question of the need of a booster dose. And once you go into that direction, then here's a question. Does a booster vaccine that has to be exactly the same as the original vaccination? And maybe not. There are very interesting works out there where for boosting immune response later in life, different strains could be potentially used in order to broaden the breadth of the protective immune response, or a more sophisticated targeted antigens could be used to type of narrow down the immune response from this global antibody response into more specific neutralizing targets, and that would increase the efficacy of the vaccine. And last but not least, we would love to have a vaccine that is more immunogenic with less number of doses. That is always a question that a vaccine developer chases to solve. And for that, maybe the current vaccine is now the path moving forward. I have to say that yes, it does work. Yes, it provides protection, but we should consider alternatives if we want to cover more diverse strains for sure.
All right, I think we have just enough time to address one more question here. That question is, might a refreshment of MMR vaccination be recommended in the elderly?
That is a very good question. And at this point, my recommendation would be yes. Yes, absolutely. Yes. But this is an area that entrenches with personalized immunity and the modern way of looking at immunizations. The current serological assays may tell you whether your immunity to measles or rubella, it's still up, and it's still standing strong as you have a more advanced age. So that's a test that can be done, and you will know whether your immunity to measles and rubella, for example, it's still there. And here's the conundrum: is that if it's not, then your way to boost your immunity will be to take the MMR or the Mmrv vaccine. So either way, you are taking, you're boosting your immunity not only against measles and rubella, but also against measles, mumps, rubella, and potentially varicella. So I would say, if you wanna do it, take that one shot of Mmrv vaccine. It's definitely now in the recommendations of the ACIP, which is the American College for Immunization Practices. The third dose is certainly an option for a population at risk, and also there is a test. There are tests, serological tests, the type Euroimmun produces and Collins runs, that would tell you whether you actually need that booster dose or not at a specific time point.
Great. Well, thank you both very much for all of those in-depth answers to these questions. We have now reached the end of the question and answer portion of this webinar. If your question wasn't addressed during the Q&A session, or if you have further questions, please direct them to the email addresses now showing on your screen. Well, thank you everyone for participating in today's conference. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen, and your participation is appreciated as it will help us to improve our webinars. Now, please join us in thanking our speakers today, Rolando Pajan and Oliver Zen Sheet. We hope you found this conference informative. Have a great day, everyone.